Welcome to Biomarkers 101, a joint webinar hosted by Sisters Hope Foundation and sponsored by Vigil Neuroscience. We are pleased you can join us today for this very special event. I am Heidi Edwards, President and founder of Sisters Hope Foundation and Cure ALSP. We are the Global ALSP Foundation. The goal of today's conversation is to support a deeper understanding for community members on the topic of biomarkers, what they are, and how they support the drug development process. Please note, we are providing the Biomarker 101 webinar for educational purposes only. For any treatment decisions, please speak to your treating physician. Today's webinar was previously recorded and will be available on the Sisters Hope Foundation website. If there are any questions after the webinar, please direct them to info@sistershopefoundation.org. We have an impressive lineup of speakers that will be sharing their experiences to aid in giving a deeper understanding of biomarkers. First, we will hear from Erin Sullivan, the project manager at Sisters Hope Foundation and Cure ALSP. She will be giving a plain language overview of biomarkers, what they are, how they are monitored, why they are important, and how they are used. We will then hear from Petra Kaufman, Vigil Neuroscience's Chief Medical Officer. She has an extensive background in clinical practice and clinical drug development. I will now turn the presentation over to Erin Sullivan.
Thank you for joining us today to learn more about biomarkers. As Heidi said, my name is Erin Sullivan. I am the project manager at Sisters Hope in the Cure ALSP division. My grandma, aunt, and mother have all lost their lives to ALSP. When my family started on this journey, I knew little to nothing about certain medical terms that are common in this disease: aphasia, leukoencephalopathy, and dystonia, just to name a few. Over the course of my family members' diseases, I've had a crash course in medical terminology. So today I would like to share a quick plain language overview of a term we hear used often: biomarker. How many of you have ever had a fever? I'm assuming every one of us has been inflicted by a fever. And to verify that we truly did have a fever, we took our temperature. Body temperature is a biomarker. Research has told us that normal body temperature is 98.6 degrees Fahrenheit. If, when we take our temperature is higher than normal, we know that something in our body is off. Maybe an infection is present. We typically treat a fever with an over-the-counter medication such as Tylenol. After taking the medication, we can monitor the effectiveness of said medication by retaking our temperature to determine if the fever is responding. If our temperature is decreasing, we know the medication's working. Body temperature is a great snapshot of what a biomarker is, what it tells us, and how it is utilized in research. A biomarker is something in the body that can be measured. Common biomarkers include body temperature, blood pressure, and heart rate. To be an effective biomarker, research must know the normal state of a particular biomarker. Biomarkers have a few different roles in research. One, they can indicate that something in the body is not working properly. And two, biomarkers can be used to determine if a treatment is effective or not. So what are some common biomarkers in the disease that brings us all together today, ALSP? A common biomarker that everyone will be familiar with is brain composition. Research has given clear indicators of what a healthy brain looks like. Research has shown and is still working on uncovering more hallmark indicators of ALSP. Some common brain changes that indicate ALSP that can be seen on an MRI are things like shrinking of brain matter, also known as cerebral atrophy, and damage to the white matter, also known as white matter lesions. I hope you found this brief layman's summary of biomarkers helpful. Now, Dr. Kaufman will lead us through a deep dive into biomarkers of ALSP, their importance, what research is learning about biomarkers, and how they're being utilized in drug development.
Thank you, Erin. Now I will turn it over to Petra Kaufman.
Thank you for the introduction, Heidi and Erin, and welcome to everyone to the Sisters Hope Foundation webinar. My name is Dr. Petra Kaufmann, and I'm the Chief Medical Officer of Vigil Neuroscience. Our goal is to discover and develop new treatments for both rare and common neurodegenerative diseases. As a neurologist, I have found myself many times in a situation where I figured out what a patient had. However, after learning about the diagnosis, when the family asked me, well, what can you prescribe? I had nothing to offer other than good care. And I went into drug development and joined Vigil to help make a difference. I understand that biomarkers and natural history studies are topics people would like to learn more about. So let's start with biomarkers. I appreciate Aaron's opening comments on the challenging of keeping up with all the language about new diagnoses, as well as important topics related to drug development, such as, the biomarkers. And in short, they are biological markers. It's signals of what is happening inside the body that can be measured. The key is that they are measurable compared to things that are not as easily measurable, such as how people functioning and feeling, which is ultimately, of course, what is more important, but hard to measure. So at Vigil, we are identifying biomarkers that can potentially show early on how our experimental treatment is affecting patients. In some cases, biomarker findings can provide information before we see clinical signals, clinical signs and symptoms of the disease. So it can be more efficient and faster in evaluating, potential treatment efficacy and safety as well. So let's talk about more specifically in the next slide, how biomarkers can help in ALSP. And there's a lot of information here, but I'll walk you through it step by step. So, we learn about biomarkers and how a particular disease progresses over time through a natural history study. So starting on the left, people get a diagnosis, genetic test, and then there are natural history studies. These are also called observational studies. That means that you follow a condition or a disease to see how it's naturally progressing with our treatment intervention, hence the name observational. Natural history studies help to build a foundational understanding of the disease. This research can then be used in future treatment development and provide benefit to the patient community as a whole. So you may be thinking, well, I already know how the disease progresses. I've seen it firsthand in my own family. But, for the doctors and experts who need to see how a treatment works, and also for regulatory agencies like the FDA, they need to understand more about how the disease progresses without treatment, so that the data can then be compared to how the disease progresses when you give a potentially effective treatment in a clinical trial, right? And so, we understand that participating in a natural history study where you don't get a potential treatment can be a special burden on patients and families, and they were really selfless in participating. But I want to give you an example from my own work, where natural history studies actually helped getting not one, but 3 new drugs for a rare disease approved in the last decade. So as a neurologist, I've seen, many babies with spinal muscular atrophy, SMA, which is a disease that leads to loss of nerve cells, weakness, disability, and ultimately shortened lifespan. And as you can imagine, I've had to have many tough conversations with parents, but we were actually very fortunate because many of the families participated and dedicated their time to do natural history studies. And these families and their sacrifices were actually pivotal in the development of a treatment for SMA. And now, a decade later, we have actually 3 treatments available, all of which use the data that were collected in the natural history study for the trial planning and ultimately the approval. So these natural history studies sort of allow us to pull back the curtain and take a closer look at early signals that may help us determine a potential treatment effect. So understanding if a potential treatment may slow disease progression in terms of clinical symptoms may take many years. Because patients don't have time to wait, we are looking for ways to understand earlier if there are signals of a potential treatment effect. And a robust understanding of such a treatment is the foundation for drug development and ultimately for the approval by federal agencies such as the FDA in the United States or the European Medicines Agency in Europe, who need to really carefully evaluate if the treatment is safe and effective before it can become available to the public, before it can be an approved treatment, which is the ultimate goal. So you see here on top of that arrow some of the steps that are needed here. And one of them is the Illuminate study, which is a natural history study for patients with ALSP caused specifically by mutations in the CSF1R gene. So I want to be sure to thank all the participants and their family members who help us collect these important data on ALSP biomarkers. And I'll dive deeper into specifics on the next slide, but at a high level, some of the biomarkers that have been evaluated in the Illuminate study are changes in the brain found on MRI scans, which are potentially related to disease progression. And these data will help us, for example, understand if we can potentially slow progression of ALSP in a clinical trial. Importantly, measuring these signs and symptoms of disease like ALSP, can take years. But if we look for changes in these biomarkers like soluble CSF1R or MRI changes, we hope to speed up that process. So the biomarker data are meant to support a phase 2 clinical trial design, which is called the IGNITE trial, that is evaluating an experimental treatment called iluzanebart, or also Vigil 101, in patients with symptomatic ALSP. And you see here sort of some more specifics on these biomarkers in the natural history study. ALSP is a disease of the nervous system. So the central nervous system is composed of the brain and the spinal cord. So we need to know what's happening in the brain. Therefore, we can use a brain image or MRI, or we can measure proteins in the fluid that surrounds the central nervous system, surrounds the brain, and that's called the cerebral spinal fluid, or CSF for short. And CSF is routinely collected by neurologists using a thin needle that's inserted in a patient's back into the spinal canal where the fluid is, and it's well below the end of the spinal cord. So the risk is actually minimal, and we use a numbing medicine to make it as comfortable as possible for patients. We realize a lumbar puncture can be difficult. However, as I was pointing out, some of the important biomarkers like the soluble CSF1R protein that's kind of linked to the disease causing gene can only be measured right now in the CSF and could not be evaluated in a blood draw. The data from this Illumina natural history study thus far really informs our understanding of how we can evaluate a potential treatment response. And we think that these changes can potentially reflect the disease progression. There's another biomarker, the neurofilament light, which is also indicative of the disease process, that we are trying to also measure in the blood, because we realize that that's easier to do, and then we can do it more often. So in the next slide, let's look a little bit more at the MRI in detail as a biomarker. So you can see here an example of how MRI helps to measure changes related to ALSP in the brain. Starting on the left, you can see an MRI image showing a cross-section of the brain. And you see the skull in a sort of lighter color at the outside, and then the brain, the gray, matter inside. And you see there in the brain tissue sort of darker areas. That's where the gray matter is. That's where the neurons are. And then lighter areas, that's where the white matter is, where all the connections are. And then right in the middle, you see two chambers that are filled with they look dark here, black, and that's where the cerebrospinal fluid is. It surrounds the brain, but it's also inside these chambers. And over time, now when you look on the image in the middle, which is taken after 6 months, and the image on the right, which is taken after 9 months, how we look at the size of these ventricles, we call these fluid chambers, increasing, which signals potentially the progression of ALSP. So with this, MRI can be a really helpful tool in assessing the progression over time, even before there is a clear change in the clinical symptoms. So let's look a little bit more, now that we've discussed the imaging and fluid biomarkers, at the actual natural history study, the Illuminate study, on the next slide. So this study is a 36-month study. It has an observation period of 3 years, where patients come to the clinic and these biomarkers are measured along with clinical tests and evaluations. The changes in brain structure, the MRI imaging, happens every 6 months. The CSF, which is taken with a lumbar puncture, is happening once a year. And then blood draws are also happening every 6 months to try to measure some of these proteins in the blood. So this is what's happening in the study. On the next slide, then, we have some additional information. The study is ongoing right now in several countries, and travel assistance is provided for eligible participants. If you think this might be of interest to you or one of your family members, the best next step is to discuss it with your doctor. And you can also find information on this and other studies on a website called clinicaltrials.gov, or you can send an email always to patients@vigilneuro.com to find more information. The data will be de-identified and treated confidentially when they're used in drug development. And in the next slide, I'll tell you sort of in summary what we found in the natural history study. And we basically we're very pleased to see that we can see changes in the biomarkers potentially earlier than changes in the clinical symptoms. The ability to observe these changes before disease symptoms manifest makes them a powerful tool in the disease characterization that is leading to drug development. And we see essentially on MRI and on some of the fluid markers potentially disease progression, and the data are the foundation not only for the community to have a better understanding of ALSP, but also as a baseline comparison for the phase 2 IGNITE clinical trials, and hopefully future research to develop new treatments for ALSP. So we're looking forward to sharing with you further updates provided by both the Illuminate Natural History Study and the clinical trial later this year. And again, we're in this together. I want to thank the patients and care partners and families who selflessly participate in this research. And with this, I hand it back to Heidi.
Thank you, Petra and Aaron, for an amazing overview of biomarkers, and thank you to Vigil Neuroscience for sponsoring this webinar. As you heard during the presentations, a biomarker is something in the body that can be measured. Tools to assess biomarker for ALSP are MRIs, blood samples, and spinal fluid samples. As Petra discussed, we look at neurofilament light, NFL, in spinal fluid as well as in blood, because the technology is not advanced enough in looking at biomarkers in just blood. Now let's answer a few questions from our community. When will Vigil release data from the natural history study? And will this happen in periodic intervals over the next couple of years, or when the study closes? Petra, would you please answer this question?
Sure, Heidi. So yes, we will share data and have done so. We had interim findings in November 23 on the natural history study, And this data was actually also recently presented at the American Academy of Neurology conference in Denver by Dr. David Lynch, who is a British a neurologist in London. And these findings are, the highlights from those findings can be found on the Vigil website in a press release, and there is a summary for you. So we'll continue to periodically share data with you. The next planned update will be in the fall of this year. And, sharing data is critical because we hope this will help the community and future clinical trials. So we are really glad that we can provide this information to the community. This process is following certain kind of specified rules and happens at certain intervals per the protocol. So you have to follow this process. But I just want to say once again, Heidi, that of course none of this would be possible without all the current, participants and hopefully future participants in the study. So thank you.
Thank you, Petra. Now that I understand the purpose of biomarkers, I would like to participate in the natural history study, but there isn't a center close to my home. Will you be adding additional sites? Petra, if you could answer that question as well.
Yeah, so thanks for this question. And it's great that understanding more about biomarkers prompts people or this particular listener to ask about this, because we definitely, as you heard, biomarkers are really helping us in developing new drugs. So Vigil is a relatively small biotech company trying very hard to make a difference in ALSP. We wish we could have clinics in more locations to make participation as convenient as possible. However, given the size of our company, this is unfortunately not possible. But please do keep in mind that the study visits are relatively infrequent, twice a year. So we understand that this is still, can be a great distance for some families to travel and significant sacrifice of time. So please know that there is travel help available. And, the study is ongoing, so we have to stop enrolling new patients this summer. However, we will continue to follow patients enrolled by that time to chart their progression. So, still open right now for participation.
Thank you, Petra. And thank you to our community for the great questions. We will see firsthand the progress of research on biomarkers through your participation in natural history studies, as Petra discussed. A natural history study shows how a disease naturally progresses by collecting important data. These are exciting times for the ALSP community, and we thank you for your support and dedication. Thank you for joining us today. We are looking forward to another webinar, Biomarkers 102, in early June. We will be hosting an expert clinician that will answer any further questions you have about biomarkers. Please send your questions to info@sistershopefoundation.org. We look forward to hearing from you and seeing you again soon.